Thank you all for joining us today on this almost holiday break. And welcome to American Floral Endowment's Grow Pro webinar series. I'm your moderator, Brian Schulker. I'm a member of the AFE's Young Professional Council and a third-year PhD candidate at North Carolina State University, studying horticultural growing media. My research encompasses the hydrologic dynamics of growing media with a focus on water use efficiency with renewable substrate materials. Today's session is on viruses in flower crops. On behalf of the Endowment, I'm excited to be a part of AFE's Grow Pro webinar series that features a new topic every month presented by an industry expert. Webinars are free to everyone thanks to the generous support of ASB's sponsors. Speaking of those sponsors, today's session is sponsored by Ball Seed and Syngenta Flowers. Ball Seed is North America's leading wholesale horticultural distributor, combining extensive experience, innovative thinking, and world-class customer service. Syngenta Flowers is one of the largest wholesale breeders of hybrid flower seed and cuttings in the world, developing and producing flower seeds and cuttings for growers internationally. If you'd like to learn more about our sponsors, or if you're a supplier and interested in becoming a sponsor for a topic, you can find that information on AFE's website at endowment.org/growpro. If you'd like to learn more about the webinar series, or if you're a supplier and interested in becoming a sponsor for a topic, you can find that information as well on AFE's website at endowment.org/growpro. We hope you're all having a great holiday season so far. AFE is getting into the holiday spirit over on social media as they're in the middle of their 12 Days of Giving Back Through AFE campaign. During the 12 days leading up to Christmas Eve, they're highlighting the many programs, resources, and opportunities the Endowment offers, as well as sharing the many ways we can give back to the industry by supporting AFE. If you'd like to make a contribution that supports more floral research funding to bring you key findings like those presented in Grow Pro, internships, scholarships, and other educational programs for our industry, visit the link in the chat to give a gift to the floral industry this holiday season. Today's session was pre-recorded in English by Dr. John Hammond. After the presentation, Dr. Hammond will join us for a Q&A. Feel free to submit your questions through the Q&A feature or chat at any time. We'll answer as many as we can before the end of the hour. Unanswered questions may be answered through a separate email exchange. This session is being recorded and will be shared to AFE's YouTube account. Through YouTube's accessibility features, you can access closed captions in other languages. To get us started, I'd like to share a bit about today's expert speaker, Dr. Hammond. Dr. John Hammond was born in Cambridge, England. He earned a bachelor's degree in agricultural botany from the University of Reading, England in 1976. In 1980, he received his PhD degree in plant virology from the University of East Anglia, Norwich, England, for work at the John Innes Institute. After postdoctoral training at Purdue University and the USDA Agricultural Research Service, Hammond joined the Florist and Nursery Crops Laboratory, USDA ARS, in Beltsville, Maryland, as a research plant pathologist. John, welcome and thank you for presenting today on viruses in flower crops. Enjoy the presentation.
It is my pleasure to talk with you today about viruses in flower crops. I'm John Hammond. I'm from the USDA in Beltsville, Maryland, and I've worked with ornamentals for a long time. So among the topics I have asked to address are the use of rapid tests for virus detection, best practices to avoid problems with viruses, Poinsettia mosaic virus, Coleus viruses and viroids, viroids in solanaceous crops, hosta viruses, and Petunia vein clearing virus. And I will conclude by discussing emerging viruses, as we will undoubtedly continue to see new viruses. Flowering plants can be affected by viruses, many different viruses. Transmission may be by insects or other vectors from the parent plant to the next generation of propagules, cuttings, or via seeds. Virus symptoms in flowering plants are often expressed as necrotic spots, stunted growth, and leaf mottling or mosaic. Infected plants often produce lower yield and quality, sometimes causing crops to be unsaleable, and also posing a threat to the health of other plants grown in the same spaces through either mechanical or vectored transmission. However, there are a few viruses and virus-like agents which can add to ornamental interest and please the customers. But plants infected by such viruses or agents may pose a risk to other crops in which the same virus may cause obvious reductions in yield or quality. Such risks should be minimized. And I hope here to identify the symptoms of viruses and methods to control the damage to plants to help you. Here are some examples of floral symptoms, a variety of crops. Angelonia, top left, infected by Angelonia flower break virus, which causes, you can see some deeper coloration and also dimpling on the petals. An orchid flower infected with Cymbidium mosaic virus, and you can see unusual coloration on the petals. A flower break on Scheisser stylus infected with bean yellow mosaic virus, and flower break on impatiens infected by impatiens flower break virus. On the lower row, to the left, gladiolus infected with bean yellow mosaic virus, one infected with both bean yellow mosaic virus and cucumber mosaic virus. A bulbous iris infected jointly with iris mild mosaic and iris severe mosaic. Tulip infected with tulip breaking virus. Kalanchoe infected with Kalanchoe mosaic virus, and a lily infected with lily mottle virus. So you see the increased intensity or break of pigments on many of those flowers. Now, there have been changes in production practices that have had a significant impact on the number of viruses that we see in ornamental crops. The trend towards vegetative propagation and the significantly increased scale of production with widespread distribution means that virus problems can rapidly become widespread. Viruses in the mother plant are transmitted to close to 100% of the propagules, and vectors or mechanical transmission can lead to the accumulation of additional viruses. Mixed infections are very common and often cause more severe symptoms than individual viruses. Here are some examples of foliar symptoms on different hosts. And one thing I would like to point out in particular here is the plants, the different leaves of Magnolia tripetala infected with apple mosaic virus. All of these leaves are from the same plant, and the differences in symptom expression are quite significant between leaves. This could be affected by differences in temperature when each of the leaves developed, by differences in the light concentration, or other factors affecting the plant development. I would note also that in this mixed group of plants at the bottom, there are green flecking on some of the leaves of this purple-leafed sweet potato. And then you have papery necrosis on, I'm not sure what this crop is down here, possibly petunia. But mixed crops and unknown viruses, some viruses can have wide host range and can be transmitted from one host to another. And in some hosts, they may cause negligible symptoms, and in other hosts, they may cause severe symptoms. So how can we deal with this? One of the major means of doing so is by careful screening, selection, and sanitation. If we start with an infected mother stock, represented here by 2 green healthy flowers and 1 red flower representing an infected plant, if we apply thorough sanitation, we eliminate that infected plant and then go forward with cutting propagation from the healthy plants. So the next generation of cutting produced plants should all be healthy. Then move on to plug production and keep up the high levels of sanitation. The crop should remain free of virus all the way to wholesale and retail with no infection among the crop. On the other hand, on the right-hand side of the screen here, without any screening and sanitation, that initial 1/3 of plants infected will continue to transmit to the same proportion through cutting propagation, but throughout plug production, There will likely be some mechanical transmission to additional plants. And then by the time you get to wholesale and retail, you will have increasing levels of infection and potentially 100% infection of the plant. And remember that also additional viruses may be introduced during the production protocol. So moving on to talk about rapid test strips. And these are wonderful tools, but they have some limitations. With exceptions, they only detect a single virus. There are some exceptions in that there are some strips that are deliberately designed to test for 2 or 3 viruses, such as some of the commercial orchid tests available, which can detect both the Odontoglossum ring spot and Cymbidium mosaic virus and differentiate between them in the same test. Some other tests have some level of cross-reactivity with different viruses. So, for example, some of the Tabamovirus tests will detect more than one Tabamovirus. And then there's the Podivirus group test, which was actually developed using an antibody generated in our lab, which can detect almost any member of the genus Podivirus. So, using yourself in-house rapid test strips, they can give you a result in as little as 10 to 30 minutes. They're usable in a greenhouse or in a lab. And of course, the results are confidential. Nobody else need know what results you get. However, there are only a limited number of rapid test strips available, and mainly only for the major viruses that infect multiple crops. And that's purely for commercial reasons. It costs quite a bit to develop the tests. They also have relatively low sensitivity compared to lab-only tests, such as ELISA or the many molecular tests. And negatives may also result if the virus not tested for is present, while there are typically no positive controls other than the control line on the test. And all that the control line on the test shows you is that the reagents are functioning and reached that control line. It's not a positive test for an infected sample. On the other hand, in commercial labs, they almost always use positive controls. The reports are confidential, provided to the people who submitted the sample. They can test for many more specific viruses. Some commercial labs offer crop-specific tests, and they also have some broad-spectrum generic tests that may detect novel viruses as well as currently known viruses. They can also offer higher sensitivity tests, ELISA, RT-PCR, and a variety of other molecular tests. They also have the ability to sequence PCR products generated using genus or family-specific primers. They can sequence those products to identify either the specific or novel viruses. The adverse point of commercial labs is that you need to ship the samples and the results are not immediate, although they come pretty fast. And they are more expensive generally than carrying out tests in-house. So here are some rapid test strips for detecting Pepper-mild mottle virus. On the top left here, I've shown samples testing various different hosts infected with Pepper-mild mottle virus, so that you can see the control line here indicated with C and the test line here indicated with T. And not all of the plants are showing clear positive samples. And then lower here, just to show you can test a large number of samples. This was a test for Pepper-mild mottle virus in 50 seeds from a pepper breeding line. And out of these 50 seeds, you can see that there were only 4 plants marked by the black dots down here, which have very weak or negative reactions. So it costs a little bit of money to test a lot of samples, but if the knowledge of whether a sample is infected or healthy is important to the development of your crop, it can well be worthwhile. This is just to show the possibility of cross detection. So these are test strips specific for Plantago asiatica mosaic virus, but it also, so here you have a healthy plant, Nicotiana benthamiana infected with Plantago asiatica mosaic virus. Here's some lily samples that are either infected or not infected. I think they all have even weak results of lilies that are infected. Here we have 3 plants of lemon balm that are infected with tulip virus X, which is the most closely related virus to Plantago asiatica mosaic virus. And you can see that there are weak results here. On the other hand, there are no results with Tricertis infected with lily virus X, which is another related virus, but not as closely related virus. And here you can see 3 samples of Nandina in which you can see some results, but the results are largely obscured by the pinkish color. So one other disadvantage of the test strips is that they are more prone to than ELISA tests to interference with the pigments from the plant tissue. This occurs especially in plants with red pigments in the leaves and stems, as is seen here by these blotchiness here of the Nandina samples. So to move on to some of the best practices for avoiding viruses. Firstly, is to get healthy material. Purchase your plants and cuttings from reputable suppliers, and if necessary, ask about their virus prevention and testing practices. Secondly, note that virus derived from tissue culture does not guarantee freedom from virus infection. In some cases, tissue culture system suppresses the level of virus replication below the level of detection of ELISA or even conventional endpoint RT-PCR. In experiments in which virus-free tissue culture derived plants were acclimated in the greenhouse and grown under isolation, some plants were found to test positive up to 3 months after removal from the tissue culture after virus levels recovered, allowing detection to occur. If possible, plants grown from new sources or types of plants not previously grown should be grown in isolation from established crops. The entry restricted to the areas where they are grown, and the crops observed carefully for possible virus symptoms before mixing them with plants in which you have seen no problems. If any suspect symptoms are observed, use available tests for the most common wide host range viruses, which include tomato spotted wilt virus, impatiens necrotic spot virus, cucumber mosaic virus, and alternanthera mosaic virus, or submit samples for commercial testing. As noted before, some of the commercial labs have specific panels of tests that they apply for specific crops. And here is an example of a virus detected in tissue cultured plants, which were presumed to be virus-free. Angelonia flower break virus infects not only Angelonia, but also Verbena, Nemisia, and annual phlox. It's often asymptomatic or with mild foliar symptoms, especially in Angelonia, where it causes some plant stunting, as you can see bottom right, the Angelonia flower break infected plant compared to a healthy plant, and obvious flower break and dimpling in many cultivars of Angelonia. There's also here some slight mottling in the leaf. But some tissue presumed to be virus-free was exported to Australia. And in Australia, testing revealed after growing the plants out that the asymptomatic tissue of Verbena is asymptomatic even after grow out, was actually Angelonia flower break virus infected, and as a result, the shipment was destroyed. More best practices for avoiding viruses. Isolation of the mother stocks, the stocks believed to be virus-free to the maximum extent possible. Those should be isolated, access limited, and entry restricted to the start of the day before visiting any other production facilities, and never proceeding to the isolated stock after visiting production greenhouses, which are more likely to have virus infection. Protective suits can be used in limited access areas, and sanitation, including hand washing and disinfectant foot baths, can be used at the entry. In all greenhouses, generally, it is advisable to exclude arthropod vectors by placing fine mesh screening over the vents and fans, and to use sticky traps to attract and capture potential vectors which do enter. And those sticky traps aid scouting and the control of infestations before they become established. I cannot emphasize enough that good sanitation is required to maintain plants free from virus or other problems. Frequent soap and water wash for hands, potentially use of disposable arm covers, treating tools regularly to disinfect them, with each worker having multiple sets to allow adequate contact time for disinfection before the next use of each tool. Disinfecting the tools and machinery between handling different cultivars or crop types. One note though, over time, tool disinfection will cause corrosion or dull blades, and routine sharpening of tools may become necessary. Here are some more examples of foliar symptoms, some ring spots on the bottom left and on the orchid here, which is caused by a tosper virus. Some ring spots and line patterns here on epimedium, line patterns on dicentra, veinal-associated reddening on nandina, and nice mosaic on a celosia infected with alternanthera mosaic virus, and mottle mosaic on verbena infected with bean yellow mosaic virus. Going back to best practices, many viruses are readily transmitted by contact. And this could be by hands, tools, machinery, or during horticultural operations, including pruning, plant spacing, or transplanting. These viruses typically affect the epidermal layers of the leaves, as well as spreading throughout, that the epidermal layer is usually the first layer infected. And then the virus moves long distance within the plant via the vascular system. Some of these viruses have no specific vectors, and mechanical transmission is their primary means of moving around. Some other viruses are mainly restricted to the vascular system and rely on vectors which feed on the vascular tissue, acquire the virus during that feeding process, and then transmit the virus by feeding on the vascular tissue of another plant. Most of these viruses are transmitted either by aphids, whiteflies, or leafhoppers, all of which feed on the vascular system, as well as in some instances on other tissues of the plant. And typically there are specific vectors for each type of virus. Sanitation and minimizing contacts between the crops will reduce any virus spread within the facility, even if you are unlucky enough to have infections. So here are illustrations of some of the things which help to control virus infection. Here you can see hand washing, sink and paper towels, foot baths, and protective suits. Especially in the highest levels of sanitation, restricted entry is strongly advisable, and the use of protective suits, either disposable or frequently laundered. Blue and yellow traps to monitor insects lead you to finding infestations at the earliest possible time and treating before they become more widespread. The yellow is typically attractive to aphids, and the blue is more attractive to thrips. Then this is one of my favorite bugaboos. Flat trimmers are used widely to trim plants to encourage secondary branching, and obtain a uniform crop. However, these might almost have been designed for virus transmission because they are causing cut surfaces on one plant, becoming contaminated with sap, and then cutting another plant, or in fact, many other plants. And I have yet to see one of these machines which incorporates a system to disinfect the blades. I have talked at a couple of trade shows to manufacturers' representatives selling these machines and have asked them why there are no facilities to disinfect the machines between samples, and they really don't seem to get the point. So I have yet to see a machine that has such decontamination built into it. If your own staff are going to do the decontamination, there are a number of ways that it could be done, using bleach or quaternary ammonia compounds, or possibly UV light, but you need to have adequate time and exposure to effect good sanitation. But even washing with plain soap and water would be better than doing nothing and eliminate some of the sap and possibility of infection. So poinsettia mosaic is one of the things that I was particularly asked to address. And this virus, or at least the disease caused by this virus, were reported as early as 1927 in Germany, and similar or milder symptoms were reported in the US in the early 1950s. And Fulton published in 1980 the first description of the associated virus, and they showed that poinsettia mosaic virus is similar to turnip yellow mosaic virus, which had been known and characterized long before. The source plants of Euphorbia pulcherrima show mainly mild foliar mottling with occasional distorted leaves and bract. However, when the virus was transmitted, either mechanically or by slip grafting, to Euphorbia cyathophora, these plants developed prominent foliar vein clearing and later mosaic patterns. Now, the poinsettia itself tend to become free of symptoms if they are grown at 24 degrees centigrade, whereas the EU cyathophora showed obvious mosaic even at this higher temperature. Fulton purified the virus, obtained a good antiserum, and utilized it in ELISA, and found that the poinsettia mosaic was widespread in most of 150 different poinsettia types they obtained from commercial greenhouses, although many of these plants showed no obvious disease. And I would emphasize that many plants infected with poinsettia mosaic show no obvious disease. So here on the left, there are symptoms of poinsettia mosaic in a couple of different poinsettias. But in most cases, poinsettia mosaic infected plants are without symptoms, except possibly for minor leaf and bract distortion. A few plants may show leaf or bract crinkling or puckering, and less frequently mottling or mosaic, as shown in the two images on the left. The symptoms tend to be more prominent under cool conditions, 18 centigrade, 65 Fahrenheit, whereas plants of poinsettia grown at 24 degrees have been reported to become free of apparent symptoms. In contrast, the other species continue to show obvious mosaic even at 24 degrees. Even where some plants show visible symptoms, plant growth is not significantly affected, and typically only a limited proportion of a crop is visibly infected under warmer conditions, and mild symptoms may have a minimal effect on crop quality and marketability. So this is not necessarily something to be too worried about. It is possible to obtain poinsettia mosaic virus-free plants. Several of the typical methods for obtaining virus-free plants, such as heat treatment or thermotherapy, meristem tip culture, and somatic embryogenesis, can be used to recover virus-free poinsettia. However, these viruses These methods to obtain virus-free plants will also eliminate the most popular free branching habit, which is characterized by weak apical dominance in producing many axillary branches, with a corresponding increase in the number of bracts. Therefore, virus-free plants recovered by these methods grow much taller with far fewer axillary branches, and this is not the habit that is favored in the trade. The explanation for this change of plant habit in virus-free plants is the concurrent elimination of beneficial infection by the phytoplasma, which is responsible for the weakening of apical dominance. And this was shown by the work of Lee et al. In 1997. So there is potential to obtain healthy plants that still have the desirable free branching habit. And this would be by using any of the previous methods for obtaining virus-free plants and screening of plants which remain symptomless when grown at 18 centigrade may identify plants free of poinsettia mosaic and could also be used to establish a new mother stock. However, if virus-free plants have been produced through specific tissue culture or thermotherapy or somatic embryogenesis, they will have lost the free branching habit. And then they could be re-inoculated with the phytoplasma obtained from a poinsettia mosaic virus-free clone carrying the phytoplasma. Such a clone infected only with the phytoplasma would need to be maintained in an area free of any poinsettia mosaic virus-infected plants, as the virus is readily transmitted mechanically between plants. And obviously, it would be quite a bit of work to regenerate plants that are firstly virus-free and then graft inoculated with the phytoplasma. So you're talking about quite a time period to develop virus-free plants which have regained the free branching habit from the phytoplasma. There's also potential for poinsettia mosaic resistance plants. It's been shown by a team in Norway that it's possible to genetically transform poinsettia by either electrophoresis or using Agrobacterium tumefaciens, and transgenic plants with resistance to mechanical transmission of poinsettia mosaic were obtained as a result. The mechanism of resistance was derived from the generation of small interfering RNAs, which are produced from a hairpin RNA transcript, which is a partially complementary RNA sequence, as shown here, that forms a hairpin with a double-stranded structure. This double-stranded structure is then cleaved by normal plant defense components into short fragments, which are then released into to produce single-stranded RNAs, which target complementary sequences for degradation. And the sequences, the short RNAs complementary to the virus strand, would then result in the plant defense systems degrading poinsettia mosaic virus RNA, and thus preventing the spread and replication of the virus. So moving on to coleus viruses and viroids. There are quite a few viruses known to infect coleus, and these include coleus vein necrosis virus, which, as the name suggests, induces venal necrosis in the leaves. This virus is transmitted by aphids. Cucumber mosaic virus is a wide host range virus which infects many ornamentals, and on poinsettia can cause mosaic, oak leaf line patterns, ring spots, and also color fading. This is also aphid transmitted. Also aphid transmitted is tobacco etch virus, which induces necrotic foliar symptoms. And then we have two viruses, impatiens necrotic spot virus and tomato spotted wilt virus, which both cause ring spots, arc-shaped lesions, and irregular line patterns, and both are transmitted by thrips. Each of these virus types can be controlled by eradicating infected plants at the earliest signs of infection and control of the potential vectors. Of some concern recently have been the coleus viroids. There are multiple viroids that are known to infect coleus, but only Coleus blumei viroid 1 is of major importance. And that's because this is not only seed transmissible at rates from 0 to 100%, depending on the cultivar and the environmental conditions, but because this viroid induces symptoms of discoloration and growth retardation in some cultivars. However, it remains asymptomatic in other cultivars, but it's also transmitted mechanically as well as through the seed. The other 4 viroids listed here are typically symptomless, but are able to be spread mechanically. But they are therefore of little commercial importance because they don't produce significant symptoms in almost all cultivars. There are also viroids in solanaceous plants, but again, these viruses produce little damage in the solanaceous plants, which are essentially in the ornamental plants, which are essentially symptomless. And these include Brugmansia, Solanum jasminoides, and Lysianthes rantanetii. The viroids concerned include potato spindle tuber viroid, tomato chlorotic dwarf viroid, tomato apical stunt viroid, columnar latent viroid, and Citrus hexacortis viroid. And these were seen of concern as a potential reservoir of viroid infections, which could infect then transmitted to tomato and potato. In some greenhouse operations in Europe, solanaceous ornamentals were grown in the same space as tomato transplants. And these viroids can all cause major disease losses in tomatoes. Initially, stringent EU regulations against viroid infections in ornamentals were introduced, but they were reduced in 2015 as there was less overlap between the production of ornamentals and either tomatoes or potatoes. Several of these viroids also infect petunia, but typically without any apparent symptoms, even at the higher temperatures which favor viroid replication. Hosta is also infected by a number of viruses shown here. Arabis mosaic, cucumber mosaic, Hosta virus X, tobacco rattle virus, tobacco ring spot virus, tobacco streak virus, tomato ring spot virus, and the 2 tospo viruses, impatiens necrotic spot virus and tomato spotted wilt virus, which are both very wide host range viruses in addition to cucumber mosaic virus. However, the most important of these is Hosta virus X. The symptoms range from being latent to causing severe mosaic and necrosis. You can also see leaf thickening, blotching, and abnormal variegation, with some leaf distortion, desiccation, and necrosis possible. It's readily transmitted mechanically, and all cultivars may be susceptible, but differ in symptom expression. Petunia vein clearing virus is relatively unusual. It can integrate into the host genome of petunia, and in fact, it is incorporated in the genome of many petunia varieties. Under most environmental conditions, the virus remains latent in the genome, but tissue culture or significant environmental stresses, especially water or nutrient stress, can activate the virus infection. When the virus is replicating, typical symptoms are chlorotic vein clearing, with sunken leaf veins sometimes becoming necrotic. You can also see leaf distortion, reduction of both leaf size and internode length, and general plant stunting. Vigorously growing plants usually show no symptoms. It's not transmitted vegetatively, mechanically, only by grafting or by vegetative propagation. So avoidance of symptoms is largely by avoiding stressing plants which may contain integrated copies of the virus. Now, emerging viruses, we can expect to see more of these emerging over time. Their origins are usually unknown because we don't usually find out about them until they cause obvious symptoms in one crop. And the crop in which they were introduced may not have shown any symptoms at all. But a single infected mother plant can result in thousands or even millions of infected vegetative plant cuttings. So one of the tricks or things we need to do is to minimize the spread and the impact. We don't want to have another impatiens necrotic spot virus emerge, which was not only very important in the ornamentals industry, but which also bled over into vegetable production. So that's one case where an ornamental virus moved to a vegetable crop, whereas more frequently, the viruses may move in the other direction. The viruses with the widest host range are of the greatest concern. Initially, they may be known in only one host, but then they may evolve to infect many other hosts and cause significant crop losses. So one example of an emerging virus is alternanthera mosaic virus, which was first reported in 1999 from Australia in a weedy plant. Within a few years, it was detected in Portulaca in Italy and creeping phlox in the USA. Since then, it's known to infect a wide range of taxonomically diverse ornamentals from at least 18 plant families, many of which are shown on the left here. It has no known vector, but it's efficiently transmitted mechanically, including during routine horticultural operations. Many hosts show obvious foliar mottle or mosaic, but some hosts show either mild symptoms or may even be symptomless. Another virus of concern in floral crops is Plantago asiatica mosaic virus, which causes major losses in cut flower lilies, with up to 80% losses observed in the Netherlands. These are the symptoms on the upper leaves and often also seen on the buds of lilies. And you can see this cart here. Those are all discard stems from from a greenhouse operation about the size of 3 football fields that I visited in the Netherlands. In the course of less than an hour and a half that I was in the facility, they were harvesting cut flowers and they threw out 4 carts piled high like this in that hour and a half. That's an indication of how much waste there is. It's mechanically transmitted and can also be transmitted through the growing medium and during bulb washing operations. Causes the rejection of many cut stems. So this virus was initially observed in a weedy plant in the Russian Far East, and then in the early 2000s in edible lilies in Japan, but it's now widespread in commercial lilies and has an increasing natural host range, including many taxonomically diverse plants. So to conclude, screening new varieties and species at the earliest stage possible, preferably in an isolation greenhouse, before mingling with other crops, and observing and retesting as necessary in later production steps is necessary to maintain healthy crops. My thanks to Terrell Nell and the organizers and sponsors for the invitation to discuss this information with you. I also thank the many colleagues, diagnosticians, and growers who have shared plants, pictures, and information with me over the years. Working with viruses affecting ornamentals and with the growers over the past 40 years has been a pleasure. It has also been a pleasure to work and share information with colleagues from other countries who also work on the viruses of ornamentals. This is, after all, a global industry. And I'll be happy to take any questions.
John, thank you so much for that presentation. It was extremely insightful. And if you've seen the chat box, people have questions. So let's kick things off with the first question here. What hand disinfectants are adequate to kill virus? Any brand or active ingredient? How long does it take to eliminate the virus in hands? Same questions for tools.
Hands is easy. Regular soap and water and a good stiff brush. So if you follow the recommendations for cleaning your hands to prevent transmission of flu or COVID, you're doing the right thing to prevent transmission of plant viruses. For tools, it's a little more complicated. there are a number of agents that you can apply. So a minimum of 10% bleach is good, but you need to allow sufficient contact time. And the longer you can leave the tools in bleach, the better. So I would recommend having multiple sets of tools, and so that you can cycle them through treatment with either bleach or with quaternary ammonium compounds. And I don't know the trade names. I'm not familiar with that. I don't deal with that on a routine basis. At least 1 minute contact time, and the more contact time that you have with the tool and the agent, the better. Also, before you put them into the decontaminating agent, wipe as much sap as you can off the tools, because the presence of organic material on the tool will reduce the effectiveness of the bleach or quaternary ammonia and mean that you'll need to change it more often. So have multiple sets of tools so that you can treat some while you're using another one. And if possible, give them 10 to 15 minutes in the treatment.
That's good advice. The hand washing thing is definitely close to home after the 2 years of COVID, that's for sure.
Yeah.
Marianne Baglos has a question. Are there any fungicide treatments for viruses?
Yes and no. There are some botanical extracts that have been reported to have antiviral effects. Some fungicides do enhance the natural plant defense systems, so that can increase resistance to viruses. On the other hand, there are some fungal infections that can actually increase susceptibility to viruses. There are a few botanicals that are claimed to either prevent infection or to aid recovery. However, not all of those claims are well supported, and I'm not sure if any of those treatments are commercially available. Neem has been claimed to have some antiviral effects, but there are a variety of neem products, and I'm not sure which one is supposed to be the best because there are several chemicals that can be extracted from neem. I'm afraid that's the best answer I can give you on that one.
Perfect. So we do have Matt McElroy has a question. Seems more like looking for a recommendation.
Okay.
Battling INSV and cut snapdragons for the past 18 months. They steam the beds between crops, spray green shield on benches, guide wires, and irrigation lines, and treat snapdragon seedlings in 288 seed tray with mainspring and drench the crop with safari and imidacloprid. I could have said that wrong. A couple of weeks after transplanting into the beds, we then spray with a regular rotation of multiple insecticides, but cannot seem to eradicate the spread of virus.
And can you remind me, the virus in question was
INSV? Yes.
Well, the vector there is thrips. So if you use sticky traps so that you know when the thrips are coming in, use fine screening on any openings to the greenhouse that would exclude thrips. Thrips, I think, are more attracted to blue sticky cards than to yellow sticky cards. What else can you do? But really, you need to keep a careful watch out for the presence of thrips and treat them as early as possible. Thrips are certainly tend to be more damaging when the plants are in flower, but if they're in the greenhouse and there's no flowering material available, they will attack just about anything.
I think everybody has had problems with thrips at one point in their production career.
I'd be surprised if they didn't.
I know, yeah. I'd be impressed. I'd have more questions. So do you have any recommendations for disinfectants for sanitizing flats in between crops?
Firstly, wash them as thoroughly as you can with just water or a little soap and water in order to remove as much of the organic material as possible. And then soak them in either 10% bleach or a quaternary ammonia solution at the level that is recommended by the manufacturer. And for the contact time that is recommended, I would, after doing a good batch of flats, I would recommend renewing the solution or at least adding more of the agent, either bleach or quaternary ammonia, in order to keep the concentration up and maintain the effectiveness. Steam heat or dry heat are effective, but the levels that are effective are likely to distort the flats. So that's something that you need to do very carefully unless you are using flats that are made of a material that is highly heat resistant.
Fantastic. So this one's actually, I'm interested in this answer as well. Can we test for tobamovirus group instead of testing for each individual one?
Yes. So at least some of the available tobamovirus tests are quite broad spectrum. They react more strongly with the specific target, but they will frequently yield positive results from a number of related viruses. So that's a qualified yes, because there are different subgroups of tobamoviruses. So the ones that are more closely, the ones that infect solanaceous crops generally have reasonable cross-reaction with some of the commercially available tests. However, the tests that are used primarily for solanaceous crops are less likely to cross-react with the viruses that infect cucurbit crops, for example. So there is a fairly wide spectrum of detection, and you can also submit samples to one of the commercial labs, and they would have other tests like a tobamovirus specific or tobamovirus generic PCR testing. But if you think that you suffer with a particular virus, you could also send a sample to a commercial lab, find out which virus it is from having them conduct tests with the multiple reagents that they have available, and determine which one is the most effective that you can use in your own hands.
And Dr. Janos Agustin had a comment on that one as well, saying they have good experience with oils as repellents of vectors like aphids and thrips. 0.5% is often used weekly, but crop tolerance is different. Making a test spray is important here. And I think that's just...
Yes, lilies in particular, I think, have some adverse reaction to some of the oil sprays.
And then this next question, or last question that we have in the Q&A right now, I feel like you could probably talk about it for a while. How big is the virus issue? Because there's so many vegetatively propagated cuttings being produced these days.
Very big. The question is, how well do the producers do their job of testing from their nuclear stocks and mother stocks? And the problem there is that the original cuttings may originate in one country, and then the rooted cuttings produced in a second country, and then young plants imported into the US. And problems can be introduced at any stage along the way. I think somebody had put in a question about buying plugs and how they could analyze that they were free of virus. I think I answered that in already online. But it's the same largely the same answer. And the other problem there is emerging viruses, because emerging viruses will typically not be recognized by the tests for viruses that are currently known to affect the crop. And in that case, if you have something that's symptomatic but is testing negative with the normal tests, it may be necessary to send a sample to one of the National Plant Disease Network labs, which have more experienced diagnosticians. And in some of those instances, samples will be forwarded to myself and my colleague Ramon Jordan if they can't immediately identify something. And we also work with some of the commercial labs and directly with some growers as well. So, but unfortunately, when you have an emerging virus, it may take quite a while for it to emerge and find a crop in which it produces obvious symptoms. So it may remain asymptomatic in the crop that it's first known in, and somebody might not think it's a problem. But if it infects another crop in which it does cause obvious symptoms, it may be much more important than previously. And alternanthera mosaic virus was a case in point for that, as was Plantago asiatica mosaic. They were both originally found in weeds, but 30 years later, they were major problems.
Well, this next one is everyone's question. Is bleach or any quat enough to kill virus-complicated virus as TLB-RFV, tomato brown rugose fruit virus?
Yes, given adequate concentration and contact time. But the Tabama viruses in general are very stable. They will persist on surfaces, and careful sanitation is very important with dealing with those. But they can be fought against, and you can win. There was a question there about how to differentiate between nutrient disorders and virus infection. The patterns are usually different, and fertilization will not usually make any difference to nutrient deficiencies. And if you have a virus-induced chlorosis and you use nitrogen fertilizer, you're generally not going to see any difference at all. You'll just be wasting money on nitrogen. So look carefully at the patterns and contact your local extension agent or one of the diagnostic companies to get you a better answer.
It's very good advice there. So this next question is in Spanish, and I'll do my best to translate. And what I believe is how to handle potavirus in a spread.
Potavirus?
Potavirus during propagation?
Those are aphid transmitted. Again, exclusion of the vectors from the greenhouse, using sticky cards to identify the presence of the aphids at the earliest opportunity, routine scouting, and treat for aphids as soon as you see them. There is one caveat there, that applying pesticides to treat aphids may cause them to fly and infect other crops other than the one that they're initially on. So exclusion is the best policy, but they may bring in viruses from outside, as well as spreading viruses around that may occur in other crops without symptoms.
And since we're running low on time, we have time for one more question. And for you, this one will be: what would you do if you have crops showing signs of a virus, but when samples are sent in for testing, they're not coming up positive for any virus?
That means you probably have an emerging virus, and that send a sample to one of the NPDN labs, or contact me or my colleague. And there are various other researchers around who are interested and have a lot of knowledge with ornamentals. But I do have a diagnostic permit to receive samples, and I'm happy to work with people, especially with emerging viruses, to help determine what they are and produce tests that would be able to then identify them and help producers generate healthy plants.
Well, there you go. If you attended this seminar, you now also have a place to send your samples to. With that, we're going to close questions. If anyone has any more unanswered questions, please send them to us. And it's in the chat there at communication@afeendowment.org. And we and John will follow up. Thank you again for joining us today for another session of AFE's Grow Pro webinar series. Join us in the new year for more great webinars with industry experts. The 2023 schedule will be coming out soon, so stay tuned to AFE's emails and social media to mark your calendars when it comes out. In the meantime, please visit endowment.org/GrowPro if you'd like to see some of our past webinar recordings and other grower-related resources and research reports available to you for free. Thanks to industry support. If you find this webinar series and AFE's other industry resources impactful to your business, please consider making a donation as we approach the end of the year. We also ask that you please complete the brief survey about today's session where you can suggest additional topics and help us continue to improve these webinars. Thanks again for joining us, and happy holidays.